Me chamo Guilmar Oliveira, eu sou a pediatra, eu trabalho em Portugal, Coimbra. I would like to thank to the organization by the invitation. It's a pleasure for me to stay here in Andalusia. Now, it's a pleasure for me to introduce our first speaker. She's Sarah Curran. Sarah Curran is a child psychiatrist. She is a teacher of genetics or molecular genetics in behavior. She works at Moseley Hospital, and now she is going to talk about genetics in Asperger syndrome. Please.
Thank you, and thank you for inviting me to talk here today. I hope you're going to understand and follow this talk. I'm trying to briefly give you an overview of the past 25 years research on the genetics in autism and autism spectrum disorders. And unfortunately, the title of my talk is The Genetics of Asperger's Syndrome and Other Spectrum Disorders, which caused some challenges for me when I was preparing this talk, because most of the work has been done in core autism. And as I go along, you'll probably see the difficulties that is posed for trying to understand the genetic basis of Asperger's syndrome. Just to give you an overview of my talk, first of all, I'm going to talk a little bit about phenotypic definitions and concepts, give you a little bit of the history of the genetic studies and the background to this work, an overview of the cytogenetics and the progress that's been made in cytogenetic techniques over the past 5 to 10 years, talk a little bit about family and twin studies, give you a summary of the molecular genetic studies and approaches that have been undertaken, and finally summarize the talk and make some conclusions. First of all, dealing with the diagnosis of autism and autism spectrum disorders, whether one looks at DSM-4 or ICD-10, similar categories are in place. And autistic disorder, as defined by DSM-4, is probably the most well-known of the group of disorders, occurring at about 13 per 10,000 children in the population. Asperger's syndrome is less common, with a conservative prevalence rate of about 2.6 per 10,000 in the population. And then the other catch-all pervasive developmental disorder category can occur in approximately 0.2 or 20 per 10,000 of the population. Rett's disorder and disintegrative childhood disorder are also part of the PDD categories, but are usually not included in genetic studies. The difficulty I had in looking at the literature was that Asperger's disorder is diagnosed as being different from high-functioning autism by the lack of delay in language development. Now, the problem is that many children or individuals with Asperger's syndrome present in the adolescence or in adulthood even. And so for a parent to try and remember the language, especially if it's an only child or a first child, or whether it was abnormal or not, is often very difficult. So there's a blurring of the categorization of Asperger's syndrome from high-functioning autism. And also in DSM-4, it's considered that Asperger's disorder should not be diagnosed if the criteria for autistic disorder are better met. So just in terms of autism spectrum disorder, we use the word idiopathic to describe a large number of cases with no known etiology. We know that environmental factors and prenatal and perinatal complications are elevated in children with autism spectrum disorders. We know that paternal age in particular is increased in the fathers of affected children. We know there's a high genetic heritability of these disorders generally. We know that epigenetic modifications are important. And so on. So in terms of the genetic research into autism spectrum disorders and Asperger's syndrome, it's been a very short history of research. It wasn't until the 1970s that people began to understand that there was a biological basis for these disorders, and up until then, a psychodynamic view prevailed. People considering that mothers of children with autism or Asperger's syndrome were cold or aloof. And it wasn't until 1979, really, that Professor Rutter at the Maudsley carried out the first twin study showing that twins sharing 100%, monozygotic twins sharing 100% of the genetics were much more concordant for autism than dizygotic twins. In the 1980s, with the development of cytogenetics, it was found that autism also occurred in known chromosomal and single gene disorders, such as tuberous sclerosis or Fragile X. And then further, in the 1990s, there were further family and twin studies, which further confirmed the idea that autism spectrum disorders were very much genetic disorders. And then further, there was the development of structured interviews and with parents and observational schedules, which helped the development and progress of genetic research into these disorders. Advances in technology have really helped this area immensely. The Human Genome Project, worldwide collaboration to sequence the whole of the genome, that's all 3 billion bases of the genome, and was carried out only in 2003. Since that, there's been a huge number of collaborations to try and find ways of looking at variation between individuals and variation that's associated with the diseases, common diseases, and more rare diseases. Even more recently, microarray technology has developed further, which has allowed us to really focus in and look at a vast amount of information on each individual and on groups of individuals, and allows us to really try and focus on the biology of these brain-related disorders, which up until then we really couldn't do because we didn't have the opportunities to biopsy brains or look at pathology in vivo. Cytogenetic techniques have rapidly improved over the last 20 years or so. Initially, the resolution was quite poor. In the 1960s, one could look down a microscope and look at aneuploidies like Down syndrome and Klinefelter syndrome and Turner syndrome. Then in the 1970s, with banding techniques, GNC banding techniques, one was allowed to perhaps differentiate smaller areas of chromosomes. And in the 1980s and 1990s, higher resolution banding, allowed one to diagnose disorders like Prader-Willi syndrome and DeGeorge syndrome, which were then found to be associated with autism in a higher prevalence than normal. In the more recent times, with oligonucleotide and CGH arrays, we're able to look at very small areas of the genome and find deletions or duplications of a single gene or multiple genes that may be causing a disease or like autism. So essentially, autism spectrum disorders in general can be accounted for by a single gene or chromosomal abnormality in 10 to 20% of cases. And in fact, this number seems to be increasing with the development of technology. There are some clinics that say more than 20% of their cases, their newly diagnosed cases, can be diagnosed as having a single genetic abnormality causing their autism. So this has changed quite dramatically. Just an overview of the autism spectrum-related genetic syndromes. The most common genetic syndrome is a duplication of the maternal chromosome, long arm of chromosome 15, which is found to be associated with an autism spectrum disorder in something like 40% of cases. And this accounts for 1 to 2% of all cases of autism. More rare or abnormalities in the chromosome, the short arm of chromosome 16, also Fragile X, Joubert syndrome, Potocki-Lupski syndrome. Okay. Shorts. Oh, right. Okay. Okay. Rett syndrome, which is a single gene disorder of the X chromosome MECP2, are all autism-associated genetic syndromes. And you can see the proportion of these patients that have an autism spectrum disorder isn't often 100%. So there is something in the other related genome that's influencing penetrance of these disorders. So when we talk about idiopathic autism spectrum disorders, we're really talking about cases in which we can't detect a genetic or chromosomal abnormality. And there have been a number of family studies. Professor Bolton carried out one of the earliest ones in London, showing that the occurrence of an autism spectrum disorder tends to segregate in families, and that family members, first-degree or second-degree relatives, have a much higher rate of these disorders than the general population. And in fact, 2 to 3% of siblings of a child with an autism spectrum disorder will also have an autism spectrum disorder. So the sibling relative risk, or the risk to a sibling compared to the general population, is of the order of 25 times higher. And there's a co-aggregation of autism with Asperger's syndrome and milder degrees of autism spectrum disorders in the same families. There have been some family studies of Asperger's syndrome carried out, mainly by Professor Gilberg and others. And again, there seemed to be high family loading, even a higher family loading than perhaps in the autism the autism families. And in fact, in the Gilberg study, it was found that 50% of boys with an Asperger's syndrome diagnosis had a paternal family history. So there's a sense that if one can refine one's focus to Asperger's syndrome, there may be, in fact, a higher genetic loading in these families. And there doesn't seem to be, even though the rates of autism spectrum disorders is much higher in males than in females, there doesn't seem to be evidence for an increased family genetic loading in girls than in boys, as one might expect. So this perhaps reflects a sort of protective factor or factors in females, which is just causing a reduced penetrance. The twin studies, as I mentioned, in autism, there are many, all showing that the concordance rate in twins that share 100% of their genome, that's monozygotic twins, is much higher, of the order of nearly 40%, than dizygotic twins. And here one can see that from the Professor Bailey's study, that when the phenotype is broadened. The definition of autism spectrum includes a wider range of behaviours. The concordance rate for monozygotic twins increases, as does that for dizygotic twins. So the heritability is maintained and perhaps even higher if one takes a broader rather than a narrow diagnosis. Again, looking at a population study of twins in a UK sample, large UK sample, again the same sorts of heritability estimates have been generated. So with monozygotic twins being much more highly concordant, several times more concordant than the dizygotic pairs. Some statistic modelling of family and twin data has occurred, and this is going back a number of years now, but it initially suggested that one might be looking at a number of loci, genetic loci being responsible in a particular case or a family of autism. And that's probably likely to be the case in these idiopathic cases where we can't find a genetic disorder. Autism traits have also been looked at in the general population, and more recently, because of the success of quantitative trait loci analysis in genetic studies, people have seen that if one can measure these traits in a population, a large population, one has a much higher chance of finding risk genes and much greater power in studies. So there's been a number of efforts to create questionnaires that attempt to pick up these traits in the general population, and these are some of them here. Looking at the childhood Asperger syndrome test within that large twin study in the United Kingdom, it's relatively normally distributed. One can see here the distribution of traits in red in the general population, and the blue dots are the scores on that scale in children with autism. And for the 3 dimensions of autism, similarly, perhaps the repetitive restricted interests are less normally distributed than the others, but there's an attempt at a normal distribution here. The twin correlations in these in this study shows exactly the same as from the categorically diagnosed clinical samples, so that the correlations for each of the subdomains, as well as for the total cast score, is much higher in the same identical twins as the compared to the non-identical twins. And in fact, the genetic overlap between these subdomains is only moderate. So it gives one the suggestion that perhaps different genes are responsible for these subdomains within the population. And this was also found in a factor analysis of the autism spectrum screening questionnaire, where 3 factors seem to best represent the data. However, those studies have been from general population samples, and the relationship between core domains in the autism spectrum in these community-based samples may not reflect what's happening in clinical samples. For example, John Constantino in the States looked at a large number of child psychiatric patients with and without pervasive developmental disorders, and using cluster analysis and also principal components analysis, could only find a single continuously distributed underlying factor. So I think the jury is still out as to whether there is a single genetic abnormality causing a single disorder which has 3 domains, or whether different genetic abnormalities are causing each of the subdomain abnormalities in autism spectrum disorders. So, as I said earlier, there seems to be a growing list of single gene abnormalities that people are finding in populations of children with autism spectrum disorders. And, you know, from what was less than 10%, perhaps 10 years ago, has now grown to 20% of all children having an identifiable genetic abnormality. And perhaps in 10 more years' time, we'll be looking at 30 to 40%. So that's quite an interesting finding, and perhaps the most remarkable and dramatic finding in the genetic study of autism spectrum disorders in the last recent period. So we're left with sort of two contrasting paradigms regarding the role of genetic abnormalities in the risk for autism spectrum disorder. First of all, you've got the common gene variant, common disorder hypothesis, which is sort of reflected in the family, twin and population data, suggesting that there are a number of complex genes inherited in families in the population which come together to produce these set of symptoms within these affected children. On the other hand, one has the rare variants common disorder hypothesis, which has been finding that particularly where one has an autism spectrum disorder for the first time developing in a child in the family, that one might be looking at single gene abnormalities causing the disorder in that child. And these may be new in the family, or they may be passed down through generations, but just not being causing symptoms in parents or grandparents. And so these two types of paradigms need different types of approaches to sort of solve the genetic underlying abnormalities. Traditionally, linkage analysis has been successful for single gene disorders like Huntington's disease, where you've got a single gene abnormality segregating with the disease in a large family pedigree. And initially when psychiatrists, geneticists wanted to look at the genetics of these highly heritable complex neurodevelopmental traits, linkage was the first sort of analysis tool they tried. And rather than looking at large pedigrees in complex disorders, one has to look at affected sibling pairs and collect large samples of families where two or more siblings or relatives, first-degree relatives within a family are affected. To date, for the past 15 years, I suppose, these approaches have been taken with autism spectrum disorders, and there have been a number of whole genome linkage studies. I mean, it's a very powerful method, but has less success with the common alleles, conferring small to moderate risk in complex genetic disorders. There have been a number of meta-analyses, or two, the most recent one in 2006, looking at 9 genome scans to date. The problem has been that all of these linkage studies have nominated almost every chromosome in the body to be associated with autism. And so the meta-analysis was trying to sort of bring the data together and combine the analysis into what might seem the best fit. And the result of that linkage or meta-analysis was that for autism, core autism as it's defined, the region on the long arm of chromosome 7 was reaching genome-wide significance. So that was the region that was looking most interesting from all of the studies, the 9 studies that had been done till that time. For autism spectrum disorder, that's a broader category of diagnosis. Regions on the short arm of chromosome 10 and 17 were the only ones that were showing genome-wide significance. And there has been a large consortium of called the Autism Genome Project, which has combined data from several groups into the largest linkage study to date, with more than 1,000 families with at least 2 affected individuals. And again, they came up with a different chromosome that was implicated, that was on the short arm of 11. So the results of linkage studies have not been robust. Some people have tried to reduce the heterogeneity in the samples by looking at subsets based on sex, language, history of regression, rigidity. And there have been some successes. The best success has been when subsets have been according to sex, and that generated a genome-wide linkage peak. However, some of the other linkage peaks have increased with trying to refine the subgroups. And there is a sort of consideration that we should be using endophenotypes in these linkage studies, and endophenotypes, as one knows, are traits that are associated with the illness, but in the general population, that are heritable, that are manifested even when the individual isn't necessarily ill, that occur within families at a higher rate than in the general population. Again, some people have started using those techniques, and there has been some success using a language as a QTL and also social responsiveness as a QTL. But however, to summarise, the results from whole genome linkage have been poor. And there have been suggestive loci on most chromosomes, and the best evidence for linkage really on 7q, the long arm of chromosome 7, the long arm of chromosome 17, and the short arm of chromosome 11. So going on to linkage association studies, whereas linkage studies can potentially detect genes of fairly large effect size, association studies have more power to detect genes responsible for perhaps just increasing the risk by an odds ratio of less than 2. And there have been a number of approaches to association using candidate genes, so looking at the biology or the understood biology of autism and selecting candidate genes on that basis and looking for variation within those genes, or selecting regions of interest from linkage, or searching the whole genome. And again, association studies, there have been lots of them, and not many have been replicated, unfortunately. Now, that may be because the sample sizes aren't large enough, but it also may be that that perhaps we've not been looking at the right candidates. But the most promising ones are here on this slide. And really the contactin associated protein on 7q is possibly one of the strongest candidates at the moment. Also candidates within the vasopressin oxytocin system, the GABAergic system, and the glutamate system. The serotonin transporter and serotonin receptor genes, which were thought to be initially potentially very promising candidates, haven't been replicated within these association studies. And in fact, it's thought that perhaps rare variants within this system is perhaps implicated in autism spectrum disorders, and that would be different, and there would be allelic heterogeneity, and therefore difficult to find association within a larger sample. More recently, there's been an effort to do genome-wide association with advances in technologies. And currently, there's a large genome-wide association consortium which is underway, and hopefully there will be results from this study within the next few months. And they're hoping to have enough power. As you can see here, the number of samples of autism cases they have selected is still only less than 700 with 6,000 controls, but they think they will be able to generate data for association with relatively low relative risks if they do find any associations. So, to summarize association studies, a minority of associations have been found with common polymorphisms that have been replicated. The majority are within tronic SNPs, so not in coding regions of the gene. And there are many genes in which there are rare variants which have been found to be associated with ASD in particular individuals and in particular families. Just repeating what I said already, the most interesting ones, as probably the most interesting one is a member of the neurexin family of proteins, mediating neuronal development and migration and dendritic orientation. A high-density SNP analysis on 17q has been undertaken, and that's on the region of the genome where sits the serotonin transporter, and no single common variant has been found. There's an interest in the GABA, ORB3, and renin genes, particularly from animal studies. So, association studies, to summarise, it seems as though rare variants are more important than previously anticipated in autism spectrum disorders. However, none of the known rare variants consistently result in autism spectrum disorder. So there is a sort of, there is not full penetrance of these single gene mutations. And so other factors are going to be implicated even in these very biologically sound cases, such as gene-environment interactions and gene-gene interactions. This is probably the most recent development in human genetic mapping has been the discovery of extensive copy number variation in the human genome. Copy number variations are segments of DNA which are submicroscopic, so they wouldn't have been picked up until the development of recent technologies for which copy number differences have been revealed by comparison of two or more genomes. And these can be deletions, duplications, inversions, or insertions of DNA. And these copy number variations usually encompass one or more genes, and collectively they include more nucleotides than all of the SNPs in the genome. So they're likely to be much more importance and perhaps more valuable in determining which genes are implicated in complex diseases. So copy number variation can disrupt gene function in many ways, causing loss of genes, functional loss, and may disrupt regulatory elements of genes. And then, on the other hand, some large copy number variations have no overt phenotype at all. So people are currently trying to work out how best to approach the analysis of these copy number variations in disease populations versus normal populations. Just to describe, and this is an illustration of copy number variation. I'll take section A here. One has the maternal. Oh dear. Or can I go back? Yes. Maternal and paternal copy of chromosomes here. And you can see as zygotes have been formed and there's been gametogenesis, here there's a de novo loss of this particular gene or site here on the offspring, as just an example of one of the many types of copy number variation that can occur. And here's just a lexicon of genomic variation. At the smallest sort of genetic variation occurs at the single nuclear polymorphism single base pair change, differences between individuals. And then there's got all this structural variation, which can be, varying from 2 base pairs right through to several thousands of base pairs, right through to the type of cytogenetic abnormalities that one can see under a microscope. So different microarray platforms have been recently used to look at CNV variation in idiopathic autism spectrum disorders. And they're outlined there. And it's been found that de novo, that's new, the first ever copy number variations, not inherited, have been found to be more prevalent in populations with autism spectrum disorder than in the normal population or undiseased population. And a couple of studies here, Marshall and Sebat, using more than approximately 500 cases with autism spectrum disorder compared to about 1,000 controls in the population, have shown that the rate of de novo copy number variation in autism spectrum disorders is between 7 and 10%, which is quite high compared to 1% in the population. And these de novo variations are much more common in simplex families than in multiplex families. That's in cases where there's only one child affected in a family, compared to the multiplex families where there are more than one affected family member. And so, in relation to these CNVs, an interesting area on the short arm of chromosome 16 has been identified by 3 independent studies. And generally, it's been found that there's an enrichment within these copy number variations for genes that have involvement in the neuronal synaptic complex, for instance, SHANK3 and the neuroligand genes. So it's pointing to abnormalities in the neuronal development, which is caused by genes within that synapse in development. Here's again an illustration of the various sorts of copy number variation. As I described earlier, the maternal duplication of 15q has been known for a long time to be associated with autism spectrum disorders, and the sort of the grey is the contribution of the CNVs to the autism in those particular cases, with the light green being other genetic influences or environmental influences. And you can see, right down at the other end, you've got paternal duplication of the chromosome 15, which is, not really associated with autism at all. The interesting thing also more recently is that these copy number variations seem to overlap with other disorders. So, for instance, these copy number variations on chromosome 1, 15, Q11 and Q13 have been found not only in autism families or cases, but also in cases with schizophrenia and mental retardation. So you have exactly the same de novo copy number variant found in 3 different disorders. So it begs the question whether one is looking at pleiotropy, or whether we're phenotyping correctly, or whether there are other factors involved. And currently there are a lot of public databases with a lot of information on these genomic variants. There's the database of genomic variants and the autism chromosomal rearrangement database. So if one logs in there, one can find interesting aspects of the genome that one might want to scrutinize in other studies. Right. There have been some mouse models of autism, Fragile X, Rett syndrome, and GRAIL2 mouse, and these have been useful in looking at social interaction and repetitive restricted behaviours. And because mice and animals are much more malleable to intervention, one can look at all aspects of the development from embryo to adult mouse, and both in vivo and in vitro analysis of the the genetic variation. In terms of hope for the future, there have been a number of very, I suppose, promising developments in Fragile X and in tuberous sclerosis, which have come from animal studies. And particularly the use of minocycline, which is a tetracycline used for acne in adolescent populations. This has been shown in animal models of Fragile X to promote dendritic spine maturation and to actually improve learning and social integration in these mice. And so there's currently an open label trial underway looking at the effects of minocycline in individuals with Fragile X syndrome to see if it improves their development. In addition to that, there have been some glutamatergic antagonist trials which have started also looking at correcting the Fragile X phenotype in individuals. And in tuberous sclerosis, the immunosuppressant rapamycin has been shown to rescue memory and learning deficits in animal models and is currently undergoing open-label clinical trials in children with tuberous sclerosis. So, Just summing up the biological themes for autism spectrum disorders, on the one hand, one has the rare gene variants causing the disorder, and then on the other hand, one has the common gene variants. And one might think of, autism as defined categorically, as being perhaps the autisms, a bit like the mental retardations, where you've got children with a very low IQ at the one end, up to normal IQ range at the other end. And perhaps in the lower IQ range, one is getting an accumulation of single gene variants causing disorders in individuals at that end. And at the other end, with the normal IQ and less degree of disability, perhaps one is looking at the common variants causing disease at that end. And the problem is with genetic research, everything has been lumped together to date, and perhaps causing too much noise to perhaps be able to define and refine our understanding. So parallel approaches are needed to identify the mechanisms in autism spectrum disorders. On the one hand, you need whole genome and pathway array-based association studies. Deep resequencing of rare variants is going to be necessary, and integrated collections and collaborations of really large, tens of thousands of well-characterized cohorts and their relatives are going to be needed for longitudinal genotype-phenotype studies. And in addition to all of these molecular genetic techniques, there's the developing world of transcriptomics, proteomics, pharmacogenomics and metabolomics, which we can help use to try and decipher the underpinning biology of autism spectrum disorders. One can think about these genetic and environmental factors influencing intrauterine and early postnatal brain development, and different genes and environments altering brain development at different times, depending on when the insult occurs or where the abnormality is. And just an overview of where variation in particular genes might have an important effect, some of them occurring and, the bottom line is that we think there's an abnormality in the synaptic function and in summary, autism and related behaviour traits are, found to be highly heritable. There's no doubt about that. Identifying the genetic basis has been challenging, and most discoveries to date failed to be replicated. But there's been some success made by incorporation of intermediate traits and endophenotypes and identification of rare variants, I think, and structural novel mutations in autism spectrum disorders. And the current efforts really are aiming to increase the sample sizes and collaborations, gathering large samples of case controls and using meta-analysis of studies. I think more attention is needed on unique families and rare variants, because they will give us a hook into the more complex genetic architecture of autism and the spectrum. So just to summarize again, it is, family and twin data studies. I guess others will be talking about this in later aspects of the meeting. Whether one is looking at genes for 3 independent domains, or whether it's looking at genes causing a singular abnormality which is portrayed in 3 different domains, is yet to be decided upon. Linkage has been useful, but with the sample sizes that we have, I feel that further investment in linkage, unless one is going to use endophenotypes and large samples of endophenotypes or subtypes, is going to be a waste of time and money. I think the large genome-wide consortia will pick up some of the complex genetic variation associated with autism spectrum and traits in the population. And I think we're going to have combined approaches with cytogenetics and microarray technologies aiding the linkage association in large collaborative cohorts. And so just to come back to why we're all here, is about Asperger syndrome, which, just to track back to the very beginning of my talk, is actually a small subtype of autism spectrum disorders, autism being probably 3 or 4 times more common, and autism spectrum disorder being, perhaps 10 to 15 times more common. So if one thinks about pervasive developmental disorders, or a diagnosis of Asperger's or category of Asperger's is actually a very small subtype. And given the recent developments in finding single gene abnormalities in autism spectrum disorders, one wonders whether one is actually dealing with the same genetic architecture. And there's been some clues to that, in that the male-female ratio is thought to be 8:1 in Asperger's syndrome, compared to 4:1 for autism, compared to 1:1 at the lower end of the autism spectrum. Some of the family history data, looking at specifically at families with Asperger syndrome, have suggested that there is a sort of greater family loading, not only of Asperger syndrome, but also of autism, of course, and of the broader phenotype in these families. And there's this, I don't know, perhaps Frankie will talk more about the cognitive style and whether that differentiates or can differentiate people with high-functioning autism from Asperger syndrome, or whether it's genetically driven or important. And then I think there are the diagnostic issues related to Asperger syndrome. Most of the large genetic studies come from the States, and the large collections have been initiated by groups in the States, and they're using DSM-4. Of course, here in Europe, we tend to use DSM-4 a bit at the Institute of Psychiatry, or half and half.
Sarah, please, 10 minutes.
Okay. But here in Europe, perhaps people are using ICD-10. And then, of course, for diagnosis of Asperger syndrome, one has the Gilberg and Gilberg criteria, and also Satmari criteria for categorical diagnosis. So I think perhaps we need to decide on what it is we're thinking about and talking about when we talk about Asperger syndrome, or whether we're happy to lump it in as a genetic relative of autism, or whether we really want to look at it independently and separately from the autism spectrum disorders. Yes, so we need to have some agreement on... I'm out again. Yeah, I think I'm back in. On the categorical definitions and phenotypic measures, we also need to have some agreement on which perhaps quantitative trait assessment tools we're going to use, so that we can have a minimum set of both categorical definitions and dimensional assessment tools that we use in the studies, so that we can do meta-analysis and compare across groups. Again, with relation to Asperger's syndrome, we're thinking specifically about treatments and development of treatments for people with Asperger's syndrome. Perhaps we need to be thinking more specifically about the genetic causes of Asperger's syndrome and what might be useful for them. And so I think we need to, perhaps rather than having sort of Asperger's subsumed within the large ASD cohorts, we need to begin to think about collecting rather more well-categorized groups and creating some sort of collaborative effort to try and decipher the genetics of Asperger's and whether it's the same or different from high-functioning autism. And this is just a recent study from Nature Genetics suggesting that the power from sampling schemes for psychiatric genetics, the power of detecting genes is much greater if one has an approach where one's subtype, and certainly Asperger's syndrome is a subtype of ASD, is well defined. So something to think about. So thanks to my collaborators at the Institute of Psychiatry and the Institute of Child Health and Guy's Hospital in London. And just to leave you on a reminder that this is supposedly Johan Gries's view of Picasso, and that, you know, much of what we see with Asperger's syndrome is individuals with a lot of, very positive qualities, and we're dealing with a very, perhaps a disorder that we can help nurture and develop into something very positive, and it's finding, what mechanisms, either pharmacological, based on perhaps genetic and genomic background, or psychological, which will help these people to develop and their full potential in the future. So thank you.
Thank you, Sarah, for your interesting communication. Now, I think that time to ask questions after opening ceremony. I think so.
Sí, le rogaríamos que no se levantara nadie, que no abandonara la sala, porque ya están las personas que van a proceder al acto de inauguración. Simplemente vamos a cambiar los carteles de la mesa.